finish up with slide 54, I think it was. So we've just been talking about genetic disorders, is that correct? Okay. So, we also talked about Mendel's laws last time. But as it turns out, there are some variations on that. So he was a smart guy, he did very good science, but he didn't have the benefit of modern scientific equipment to do more in-depth studies that might have made his laws a little bit more robust. So there are a few things that don't necessarily adhere to Mendel's laws. So in general, they explain most of the way inheritance passes from generation to generation. They're very good about predicting proportions of certain genes in each generation that can be predicted using the laws of probability. We see that with the Punnett square, our example there being 100% heterozygous purple individuals in the offspring. And they're generally valid for all sexually reproducing organisms. However, they do fall short in explaining some patterns of genetic inheritance. There are a few things that don't necessarily follow all points of Mendel's laws. Sometimes things just get more complex. So whenever we're looking at something that is a trait that has two distinct forms controlled by two distinct alleles with complete dominance, then Mendel's laws always hold true. But there are some cases where that's not necessarily true. So if we go back to the pea plant experiment, the offspring in the F1 generation would always look like one of two parental varieties, no matter which genotypes we were crossing. There was never anything coming out of that hybridization that didn't look like one parent or the other. So that's true of all those characteristics, whether we're talking about seed shape or flower color or seed color. So even in the heterozygotes, the dominant trait was always completely expressed. So this phenomenon has later been come to be called complete dominance. So in the case where the dominant allele has its complete effect in the heterozygote, that is called complete dominance, and that is usually the case. So when you have complete dominance, that dominant allele will completely mask the recessive allele and will be expressed fully. But there are some really interesting cases where heterozygotes end up exhibiting an intermediate phenotype. Can anyone guess what this is called? Not blending, although it does resemble what you'd expect from the blending hypothesis. We call this incomplete dominance. So if the dominant allele does not express its full effect in the heterozygous offspring, the dominance is not complete, so we call it incomplete dominance. So the results do actually look a lot like what would have been predicted by that old, archaic, erroneous blending hypothesis. But because the genes themselves aren't actually getting mixed, we still know that that is not true.
Is that why we see people with eye color that looks like it's not one of the 3' ones?
So eye color is one of those things that's just, it's controlled by a whole bunch of genes, so it's not quite that simple. That's the same reason why, you know, children of parents with really different skin colors will usually have a fairly intermediate skin tone. It's not incomplete dominance, it's the fact that it's controlled by a whole raft of genes. Same thing with height. But you don't always see an intermediate phenotype in cases of polygenic inheritance either. Like, I'm a good example of that. My dad's 5'8", my mom's 5'5", and I'm 6'2". So I'm not in between my parents, even though that does often tend to happen. So about incomplete dominance. The most commonly used example of this in Bio 100 classes is the snapdragon flower. So snapdragons have two alleles, one for red and one for white. If you have a homozygous or true breeding red parent, and you cross that with a homozygous or true breeding white parent, by now you guys should be able to tell me that all of the offspring will be heterozygous, right? Whenever you cross homozygous dominant with homozygous recessive, 100% of the offspring are heterozygous. However, instead of exhibiting the dominant phenotype, like the pea plants do, in this case, the heterozygotes will exhibit an intermediate phenotype. They will be pink, not red or white, but a mix of red and white. So this is a case where the dominant allele is not completely dominant. Now, as I have casually alluded, this may seem like this is in support of that old blending hypothesis. But remember that a key component of the blending hypothesis was that the heritable factors or the genes or whatever you want to call them, whatever they thought they were back then, would be inextricably mixed, like pigments in paint. But if you take that F1 generation, which is going to be 100% heterozygotes exhibiting that intermediate pink color, and you cross those, you end up with red and white individuals again. So one of the cool things about incomplete dominance is it really allows you to conceptually visualize the genotypic ratio. So when you have complete dominance, if you cross two heterozygotes, you end up with one homozygous dominant, two heterozygous, to every one homozygous recessive. In the case of those pea plants that exhibit complete dominance, the phenotypes are 3 to 1. But in this case, your phenotypic ratio is the same as your genotypic ratio, which is kind of convenient. Now, obviously, the blending hypothesis is incorrect because the genetic potential to produce red and white flowers was not mixed, was not blended to produce those pink flowers. They just acted in consort with each other and can later be separated in future generations. Because the alleles that produce red or white flowers will segregate during candy formation. So as with any cross between heterozygous parents, you have that 1 to 2 to 1 genotype ratio. And here we see that as a 1 to 2 to 1 phenotype ratio as well. Did anyone miss the question on what is genotype and what is phenotype on the quiz today? Okay, I'm going to explain that again because I want to make sure everyone is completely solid on this. So an organism's genotype are the alleles it possesses. Generally, we're talking about genotype in the context of one particular character. So it would be the alleles it possesses for that gene. In this case, big R, or homozygous dominant is the genotype of this individual. Now, the phenotype is the physically expressed trait that that individual exhibits. In this case, if it's homozygous dominant, it has 2 alleles for red flowers, it will produce red flowers, and its phenotype will be red flowers. So remember that the genotype is what's in the gene, the phenotype is what's physically expressed. I 100% guarantee you that will be on the exam next week. Benefit of coming to class. Now, we also see examples of incomplete dominance in humans. One example is a crazy disorder called hypercholesterolemia. It's a big, long word, but if you break it down into its root words, it's easier to understand. Hyper meaning what? High. Cholesterol, you guys all know what cholesterol is. Generally, any condition ending in emia is something to do with blood. So hypercholesterolemia means you have higher than normal levels of cholesterol in your blood. So hypercholesterolemia is another instance of incomplete dominance. The normal state is dominant in this case. So if you have 2 copies of the dominant normal allele, then you will produce the normal amount of LDL receptors, which remove low-density lipoproteins from your serum. If you have the intermediate genotype, you will still produce some, but not the normal amount, and you will have higher than normal levels of LDL, which is the quote-unquote bad cholesterol. If you are homozygous for hypercholesterolemia, with the recessive alleles, then you will produce no LDL receptors, or effectively none, so few it doesn't matter, and you will have insane levels of LDL in your blood, and you will usually die of a heart attack by age 2 or 3. Yeah, crazy stuff. So although it is a recessive allele and the normal allele is dominant, you still see the incomplete dominance.
Sorry.
Yes.
LDL receptors, what's its role?
So LDL receptors remove LDL from your blood. LDL being low density lipoproteins. So that's the large molecules that contain cholesterol. So people that are heterozygous for this exhibit an intermediate phenotype, usually exhibiting serum cholesterol at about double the normal level. Highly prone to atherosclerosis, which is the technical term for the condition when plaques start to form on the inside of your arteries.
So if the cell removes the LDL, the cholesterol will not be up?
Yeah, if you have the normal number of receptors, it's always normal to have some LDL in your blood. That's totally normal. But when you have too much, that's when you can become more prone to other problems.
The other one's HDL, right?
Yeah. You know what HDL stands for? So LDL is low density, HDL is high density.
HDL is good for you.
Yeah, so the good cholesterol. I mean, we're sort of slowly discovering that a lot of what we thought we knew about cholesterol isn't necessarily correct anyway. It's now seeming like refined sugar is a bigger risk factor in heart disease than cholesterol is, and dietary cholesterol is probably not even an issue, which makes sense because the amount of cholesterol you can possibly eat in a day is so minuscule compared to what's naturally produced in your body already. But I'm not a doctor, so don't take any medical advice from me. Now, the condition, as I mentioned, is more serious for people that are homozygous for the disorder. They tend to have serum cholesterol levels up to 5 times higher than normal. And as I mentioned, they may die of a heart attack as early as age 2, which is really sad. So this is just an interesting example of incomplete dominance yielding intermediate phenotype in humans. So not something like skin color or height or eye or hair color, rather an actual case of direct incomplete dominance. I put way more slides in here than I need to on this edition. Yeah, I already explained that. Okay, so that's hypercholesterolemia. All right, so far, we've been discussing inheritance patterns involving only 2 alleles per gene. For example, hypercholesterolemia, or flower color in pea plants, or even flower color in snapdragons. Most of the time, though, a lot of genes in natural populations will have more than 2 alleles. Now, does that mean that any individual in that population will have more than 2 alleles for that gene? Someone be bold and take a guess.
What's the question?
So in the case where there's more than 2 alleles existing in a population for a certain gene, is there going to be an individual that has more than 2 alleles for that gene?
Yes.
No. Because remember that any individual is only going to have at most 2 different alleles for any gene. Because unless you have some chromosomal anomaly, you only have 2 copies of each chromosome. So maximum 2 alleles.
That's only for diplets, right?
Yeah, which is the majority of sexually reproducing organisms. However, you still have many different alleles in the population in a lot of cases. And a really fun example of this one that fits humans is our blood group phenotype. Who in here knows what their blood type is? Wow, really? Like 2 of you? Have you guys never donated blood?
That's right.
The life you say it could be your own. Okay. Anyway, there are 3 different alleles for blood group in humans. You guys have at least heard of blood type, right? You're familiar with what it is? Or, well, you're probably not familiar with what it is, but you've heard of it. Alright, so the 3 alleles are IA, IB, and little I. The 2 first I's are capitalized because those are dominant alleles. The last one is lowercase because it is recessive. So various combinations of these 3 alleles produce 4 different phenotypes. Now in this case, we don't have incomplete dominance. We have another really cool genetic phenomenon that is an exception to Mendel's laws, something called codominance. So in the case of human blood group, the 4 possible phenotypes are type A, type B, type AB, and type O. You've probably also heard of people being O negative or O positive. That has to do with the rhesus factor, which is independent of these alleles. So we're not even going to go into that right now. But looking at human blood type, has anyone ever actually wondered what type A or type B or type O means?
Yeah, I wondered.
Great, at least somebody has. So that refers to the presence of carbohydrates on the surface of your red blood cells. Carbohydrates are basically really big sugars. So a person that has type A blood will have red blood cells coated with carbohydrate type A. Predictably, a person with type B blood will then have red blood cells that are coated with carbohydrate type B. How about somebody with type AB blood? What are they going to have? They're going to have both. So this is a case of codominance, where you have 2 alleles that both get expressed. They're not blended, they're both expressed. So instead of having some intermediate type of sugar that's a combination of both A and B, they will actually just have carbohydrate A and carbohydrate B next to each other on the surface of the red blood cells. How about a person with type O blood? Yes, neither. So a person with type O blood has no sugars on the surface of the red blood cells. Who's the universal donor? Oh, and I'm going to explain why, in case you've ever wondered. So when you're giving someone a blood transfusion, matching blood types is absolutely important. If you give someone a type of blood that they cannot accept, it can kill them. So if a person ends up getting a transfusion with blood that contains any type of carbohydrate that's not naturally present on their own blood, their immune system recognizes it as foreign and will attack it. This is done producing antibodies that will bind to the foreign carbohydrates, which causes the donor blood to clump together and can absolutely be fatal. So for instance, let's say you've got someone who has type A blood. You give them type O blood, what's their immune response to it going to be? Nothing, because there's no sugars on the surface. So the immune system isn't going to recognize it as foreign blood, and it's not going to attack it. How about if you give them type A blood, type they've already got? So in that case, the immune system is going to recognize the sugars as something that's supposed to be there. And they'll be like, okay, this is fine, this blood is cool. But if you give them type B blood, now you've introduced blood that has a foreign sugar type on the surface, and the immune system freaks out and tries to kill it. Does the same with type AB blood. Even though those cells have the native A sugars on it, they also have a foreign sugar, so they're recognized as foreign blood. So in this case, it's not the presence or absence of the native sugar that determines the immune response. It's the presence or absence of a foreign sugar. So that's why you can give type O blood to anybody. And that's why people with type AB blood can take anything. Because they have all the sugars all the time anyway. So nothing gets recognized as foreign. So O is the universal donor. AB is the universal receiver, yes.
In the process of synthesizing blood, is the carbohydrates being attached like separate from the blood cell being synthesized? Or does it happen at the same time?
I actually don't know. I would assume it happens at the same time. So blood cells are produced in the marrow, and I would assume before they leave the site of production, they have the sugars on. I don't know that for a fact, but I'd be surprised if that's not the case. So obviously a person with type A blood produces anti-B antibodies. A person with type AB blood produces neither. They can take any types of blood. Now a person with type O blood, even though they're good enough to produce blood that can be given to anyone, they can only accept their own blood type. That's the only blood type that can only accept their own blood type. Because they produce both anti-A and anti-B antibodies. I'm type O, so I can give my blood to anybody in this room. However, most of you guys have no idea what your blood type is, so I would not accept blood from any of you. You could kill me, for all I know. If you ever want to know what your blood type is, just go drop into the next blood drive you see and donate. They will tell you. It's also a free HIV test, because they have to test the blood for HIV, and they legally have to inform you if it's positive. Although free HIV tests are everything. Anybody in here not know if they're HIV positive? Anyone in here not been tested for HIV? You've all been tested? I don't believe you. You don't know your blood type, so I bet you don't know your HIV status either. Anyway, so the 4 blood groups result from various combinations, get tested for HIV. Various combinations of those 3 different alleles. So you have those 3 alleles, IA, IB, and I, where I represents an enzyme that will add carbohydrates, A or B, A and or B, blood cells. Each person gets one of those alleles from each of their parents, like with anything else. Because there are 3 alleles, that means we have 6 possible genotypes. So we have 4 phenotypes, we have 6 genotypes. Because if you inherit one allele for A and one allele for O, what's your blood type? A, because that A allele is dominant over the O allele. Same thing with the B allele. So if you're heterozygous BO, you have type B. So both the A and B allele are dominant over I. So if you are homozygous or heterozygous for A or B, that will determine your blood type. The only possible way to get type O is to be homozygous recessive, like with any other recessive trait. I've already mentioned that type AB comes from having an allele for type A and an allele for type B. And this is a genetic phenomenon that I've already told you is called codominance. Make sure you remember what that term means. Also, be sure you remember how codominance is different from incomplete dominance. It's not an intermediate phenotype, it's both phenotypes at once. So both alleles will get their full expression in someone heterozygous for A and B.
Are there any other common examples of codominance?
Yeah, a really cool one, actually a really cool one that's an example of both codominance and a sex-linked trait is fur color in cats. So generally house cats are either black, white, or orange, for the most part. Turns out that pigmentation is usually carried on the X chromosome. White is the recessive form. Both black and orange are dominant over white and co-dominant with each other. So has anyone ever seen a calico cat? What's a calico cat? Has splotches of black and white, and orange, right? Yes. Oh, yeah, they're cool. Now, if you see a calico cat, you know it's a female. Because they have to have two alleles for color, which means they have to have two X chromosomes. Now, what determines which parts of the cat gets which color is X inactivation. So the different X chromosomes, remember that when you have two X chromosomes, one is always going to be inactivated. So the different parts of the cat, the orange part, that means the X chromosome carrying black has been inactivated. The black parts, that means the X chromosome carrying orange has been inactivated. How cool is that? Sex-linked traits are also really interesting, which we'll get into later. But there's another cool example of codominance. So codominance involves the expression of both alleles at once, rather than the expression of a single intermediate phenotype. That's how it differs from incomplete dominance. You guys ready for another big science word? Pleiotropy. So everything we've looked at so far, all of these examples, have just been cases in which each gene specifies only one hereditary character. So we've got a gene for flower color in our bee plants. We have a gene for seed color in our bee plants. But that flower color gene only specifies flower color. In most cases, though, one gene will influence multiple different characters. Genetics is unfortunately really common. So that is known as pleiotropy. I introduced an example of pleiotropy in the last lecture without actually telling you it was an example of pleiotropy. That was when I was talking about sickle cell disease. Everybody remembers what that is? That's that hereditary disorder that's really common in people from tropical Africa. Well, if you don't remember what it is, I'm about to talk about it in detail, so lucky you. So sickle cell disease, the direct effect of it is to make red blood cells that produce abnormal hemoglobin proteins. Hemoglobin proteins are what make up red blood cells and help to allow them to bind oxygen. What's going on up there?
Cheer.
You remember the rules about food in class?
Cheer.
I'm happy you're sharing.
No, I'm not eating. I was having some skills. You asked me to do some skills.
But okay, now you have the skills and you can enjoy them in 30 minutes. All right, so hemoglobin is the protein that allows your red blood cells to bind with oxygen. So if you have a disorder that causes your red blood cells to produce abnormal hemoglobin proteins, that's when you're really binding with oxygen. Another effect of it is that these molecules will tend to link together and crystallize, especially when the oxygen content of the blood is lower than normal. So when the hemoglobins are bound to less and less molecules of oxygen, in the case of sickle cell disease, they will have a higher tendency to bind to each other and form crystals. As those hemoglobin proteins crystallize, the normally sort of disc or donut-shaped red blood cells tend to deform into a sickle shape. You guys know what a sickle is? Yeah. So something used to like cut meat to curved blade. So sickled red blood cells look really weird and often look like an actual sickle blade. So as you might imagine, the body is capable of recognizing that these are not normal red blood cells. and it responds by destroying them. What do you suppose this results in? Anemia. A lack of red blood cells. This is another thing they'll tell you for free if you donate blood, is whether or not you are anemic. Because they have to test to make sure you have a normal number of red blood cells. Yes?
Does the red blood cell has the organelles inside? Like
So
cornea?
red blood cells are, even calling them a cell is almost not correct. So a lot of people wouldn't even refer to red blood cells as cells. But they generally don't have a lot of the normal things we find inside our cells. Also, as you might imagine, because of those weird jagged shapes, they do not flow smoothly through the body. They have a tendency to accumulate. And in narrower blood vessels like capillaries, they have a tendency to cause blockages, which can end up leading to severe damage to organs, including the heart or the brain. So if you have sickle cell disease, it's pretty serious stuff, and it can often be fatal. So this figure on the right here shows a nice ordered cascade of symptoms. So usually, people who are homozygous for that recessive allele will suffer from the disease. They will have severe sickle cell disease. Heterozygotes are generally healthy, though they may experience some symptoms under extreme conditions. So extreme conditions being like, if you're heterozygote for sickle cell disease, don't go climb Mount Everest. If you have really low oxygen content in your blood, that can trigger a lot of these effects. Now, symptoms can be treated with blood transfusions. Make sure you have the right donor blood type up. And there are also certain drugs that can help, but there is no cure, and sickle cell disease is often fatal. It's estimated that it kills about 100,000 people in the world every year. However, as I mentioned last time as well, there's a reason why this allele is so common. In fact, it is the most common inherited disorder among anyone of African descent. In America, it affects about 1 in every 400 people whose ancestry comes from the African continent. About 1 in 10 Americans with African ancestry is a carrier, which means they're a heterozygote for that recessive disordered allele, but they appear healthy. It is extremely rare among anyone in America that doesn't have any African descent. Very highly correlated with African ancestry. And the reason, as I mentioned last time, is because having a copy of that allele confers resistance to malaria. Malaria being an extremely serious problem in tropical Africa, kills a lot more people than sickle cell disease does every year. Anybody know what causes malaria? No. Mosquitoes transmit malaria, but they do not cause it. Malaria is caused by a tiny parasite. It's called Plasmodium, and it lives in your blood, which is how you get it from mosquitoes. So I always hate when people say mosquitoes are the most dangerous animal on earth because they kill more people than any other animal. It's not the mosquito that kills you, it's the Plasmodium. Anyway, when a person who's heterozygous for sickle cell disease is infected with Plasmodium, and when that tiny microscopic parasite enters their red blood cells, that will trigger an immediate sickling of that red blood cell. And that red blood cell will be destroyed by the immune system with the parasite going down with it. So that's how having one copy of the allele actually confers resistance to malaria. So in that case, if you live in a place where malaria is a really common cause of death, this is going to give you a much better chance of survival. That's why it's so common in people whose ancestry comes from Africa. So there's an example of pleiotropy, where a single gene causes multiple effects, affects multiple characters. That's pleiotropy. I'm repeating this so it hopefully sticks with all of you guys. Anyone have any questions about what pleiotropy means, how it works? Everybody's going to be comfortable providing you with an example of pleiotropy if I ask you to on the exam. Or a definition of pleiotropy.
Could you repeat the definition?
So the definition of pleiotropy is a single gene that affects multiple characters. Or if I provide you with the definition on the test and ask you what the term is, you'll be able to do that as well.
Could you clarify the different, the different characters?
Yeah, so in this case, the two characters that are influenced by this gene are sickle cell disease. So the more directly it's the production of normal hemoglobin. The other character is resistance to malaria. So this is an example where a single gene can affect two different characters. Whereas with that, the opposite of that, non-pleiotropy is having a gene affecting only one character, like with the flower color in pea plants. The flower color gene is never going to affect anything but flower color. That's non-pleiotropy. Normal. Did I make that more or less confusing? Less? Okay, good. Anyone else have questions on it? Okay. Then we get to see something that is basically the opposite. Polygenic inheritance. So when you have one gene that affects multiple characters, that's pleiotropy. But the converse is also quite common, which is a single character being influenced by many genes. Polygenic inheritance is probably an easier term to keep straight in your mind, because polygenic obviously means many genes. So when we're talking about polygenic inheritance, and I've alluded to that several times, talking about things like height or skin color or eye color, that's when you have your phenotype derived from the additive effects of two or more genes. So that single phenotypic character, be that skin color or hair color or whatever, is derived from the additive effect of multiple different genes, two or more. Now this usually tends to result in a character that varies along a continuum in a population. So skin color being a great example. Skin color varies in a continuum in basically any human population. It's not like you only have 6 different discrete skin tones. You can be any shade from super light to super dark. Height is another great example. It's not like everyone in the world is either 5 foot, 5'6", or 6 foot. You can be anything in between, or above or below that as well. So we're going to look at a hypothetical example here. We're going to assume the human skin color is controlled by 3 genes that get inherited separately on separate chromosomes. In reality, there's probably at least 3 genes that determine skin color, likely more. So in our hypothetical situation, the dark skin allele for each gene is going to be incompletely dominant over the light skin allele. And each of those will contribute 1 unit of skin pigmentation or darkness to the offspring. So if you're homozygous for the dominant in all 3 loci, then you would have 6 units of darkness. So someone who's big A, big B, big B, big C, big C will be extremely dark, whereas someone who has the recessive allele for all of those genes will be very light. Someone who's heterozygous at all 3 loci would be an intermediate. And because these genes have an additive effect, any other combination of free dominant and free recessive alleles would produce the same thing. So someone who's homozygous dominant in the first, heterozygous for the second, and homozygous recessive at the third would have roughly the same skin tone as someone who's heterozygous at all three loci. You guys still with me? Great. Now let's consider a cross between someone who's homozygous dominant for everything and someone who's homozygous recessive for everything. I told you guys last time that Punnett squares can get out of hand real quick when you start looking at more than one gene locus. This is going to be a fun example of that. So what do the offspring look like in this case? How many possible genotypes and phenotypes are there? This part isn't tricky. So when you're crossing anything that's homozygous dominant and anything that's homozygous recessive, you end up with all heterozygotes in the F1 generation. So in this case, all of their kids would have an intermediate skin tone. Now, if you cross that F1 generation with each other, which is one of the many things you cannot do in human genetic research, you can't force brothers and sisters to mate. What do you think the F2 generation is going to look like? So it's going to be a little bit more complicated. So in this case, we have 64 possible genotypes yielding 7 different possible phenotypes. So this is an example of a tri-hybrid cross. When you're looking at 3 different gene loci, the Punnett squares get really out of hand really quickly. Which is why I'm never going to make you guys do one of these on a test. I'm just using this to illustrate the concept of polygenic inheritance. And hopefully to answer someone's intellectual curiosity about why there are so many different skin tones. Now, in reality, human skin tone obviously has way more than 7 different variations. But this is a conceptual example of how polygenic inheritance can produce sort of a continuum of different phenotypes.
We don't even know how many alleles are.
No, I don't think we know for sure how many of those affect skin color. But does everyone understand the concept here, polygenic inheritance? Everyone's really comfortable with that? The difference between this and pleiotropy? Comfortable with that as well? Pleiotropy being one gene affecting multiple characters, polygenic inheritance being multiple characters, multiple genes affecting one character. Hopefully that mistake I just made you repeat it. So gene linkage. I very briefly alluded to this in the last lecture as well. This being an exception to Mendel's law of independent assortment. You guys remember what the law of independent assortment means, or is?
Alleles separate independent.
Yeah, so the law of independent assortment basically is what stated that the gene affecting flower color is not going to be in any way linked with the gene affecting seed color or something like that. They're inherited completely separately. However, that is not always the case. It makes perfect sense that if the 2 genes are located on the same chromosome, it's likely that they're going to be inherited together, right? Because it's much less likely for genes on the same chromosome to be separated during gametes formation. Can happen through crossover, but it is unlikely. Now, this seems inconsistent with Mendel's laws. However, somewhat ironically, this was actually documented during his experiments with pea plants. Crosses between double heterozygotes for flower color and pollen shape. So in this case, if you have individuals that are heterozygous for the gene coding for flower color, either purple or white, and the gene that's coding for pollen shape, which can be either long or round, you end up seeing clear evidence of gene linkage. If the law of independent assortment had no exceptions, then the predicted numbers of each offspring would look like this. But in reality, they had a much different ratio. So the individuals here that are heterozygous will exhibit both dominant phenotypes, which means purple flowers and brown pollen grains. But you end up seeing a lot less where they actually tend to be separate. I'm not sure why that image says red. It should say white. White flowers, not red flowers. I didn't make that. Textbook publisher did. Take it up with that. So if you look at just one of the two characters, you find here that the dominant and recessive alleles segregated as Mendel's laws would predict. But if you look at both characters at once, you don't see that 9:3 to 3:1 ratio you would expect if these characters were separating independently. Two of the phenotypes were present in much higher proportions than predicted and two in much lower. And this is due to this phenomenon of gene linkage. So gene linkage is when genes tend to be inherited together because they're on the same chromosome. This is an exception to Mendel's law of independent assortment. This doesn't mean Mendel was a moron, who did not have, no one knew the chromosomes existed yet. So it would have been really hard for him to actually get this right. Anyway, so we have gene linkage, which is chromosome or genes on the same chromosome. Are those genes always going to be inherited together? With zero exceptions. No. Because they can be separated during crossing over. Hopefully by this point everybody's familiar with what crossing over is. That's when chromosomes will actually exchange pieces of themselves with each other during meiosis. Is it meiosis 1 or meiosis 2? Meiosis 1, how do you know? Because crossing over only happens between non-sister chromatids. Because sister chromatids are identical, so crossing over would be a waste of time. That'd be like buying $1 for $1. So it will only happen during non-sister chromatids. And non-sister chromatids are only in contact with each other during meiosis 1. Remember, that's when they align as tetrads or two pairs of non-sister chromatids on the metaphase plate. After those tetrads get separated into two separate pairs of sister chromatids in anaphase 1, those non-sister chromatids will never see each other again. So if they want to trade any baseball cards, they have to do it in meiosis 1. Crossing over obviously produces new combinations of alleles that never existed before. We have brand new recombinant chromosomes. And this is how genes can get separated when they are on the same chromosome. Is everybody on board with this? Everybody still with me? Okay, great. So as it turns out, the probability of this happening, not surprisingly, is related to the locus of each gene relative to the other. Remember that a gene's locus is its physical location on the chromosome, always going to be in the same spot on the same chromosome forever to the other side. The closer genes are to each other on the same chromosome, the less likely they are to be separated by crossing over or by recombination. That's because the less space there is in between these genes, the less probability that the crossing over event is going to occur in that space. Doesn't always happen in the same place every time. In fact, this is actually how we know the loci of many genes on chromosomes. We can use their recombination frequencies to calculate how far apart from each other they are. So, hypothetical example here. We have these three genes, G, C, and L. The probability of a crossing over event occurring between G and L is 17% because they're pretty far apart. We've got C here towards the middle. So the probability of something happening between G and C or C and L is much less because they're so much closer. Does that make sense? Yes, okay. Somebody said yes. I'm assuming that's a tacit affirmation from the rest of you since no one said no. All right, ready to move on from gene linkage? We should have just enough time to finish this lecture and then be done a couple minutes early. Sex-linked genes. So besides determining sex, which is one of the main functions of sex chromosomes also contain genes for other things. Mostly the X chromosome, not so much the Y, because the Y chromosome is tiny. But any gene located on a sex chromosome is called a sex-linked gene. And because that human Y chromosome is such a tiny nub of a chromosome, it carries hardly any other genes, and almost every sex-linked gene is carried on the X. I can't actually think of an example off the top of my head of a sex-linked gene on the Y. There's probably something. But because of this reason, sex-linked traits in humans tend to affect mostly males. Great example here being color blindness. Is anybody in here colorblind? Have any of you actually been tested for it? Okay. I used to teach a lab at a different school where when we did the lab on Mendelian genetics, I would give everybody a color blindness test. I actually did inform several of my students over the years for the first time that they were colorblind. They had no idea. But it tends to be much more common in males because the gene for color blindness tends to be carried on the X chromosome. Hemophilia is another example. You guys know what hemophilia is? It's a blood clotting disorder. So people with hemophilia, their blood doesn't tend to clot properly when they get injured. So they bleed a lot more than normal people do. So part of the reason for this is that a lot of sex-linked disorders or sex-linked traits in general tend to be recessive. So if you're a female, and you have two alleles, you will rarely end up expressing that trait. If you're a male, though, you don't have another allele to dominate. So if you get one copy of that recessive allele on your one X chromosome, there you go. You're colorblind, or you're hemophiliac. A famous example of this in history is with the Russian royal family. Back when Russia had a royal family. So I already mentioned that hemophilia is caused by a recessive allele carried on the X chromosome, which is why it's seen almost exclusively in men. If you look at the Russian royal family, which you will notice in only a couple generations traces right back to the English royal family, because all the royal families were incestuous and marrying their cousins because they could only marry royalty. You see, the Queen Victoria had one copy of that allele, but because it's recessive, she was not a hemophiliac, she was just a carrier. She gives it to her daughter Alice, who's also a carrier. Gives it to her daughter Alexandra, who marries Tsar Nicholas II of Russia. Tsar Nicholas wasn't a hemophiliac, but because his son got that one recessive allele on the X chromosome that he got from his mom, he wasn't a hemophiliac. So that's how sex-linked traits work. It is possible for women to be affected by these traits, but in that case, they would have to get an X chromosome carrying that recessive allele from each parent, which is much less common. Okay, does anyone have any questions? All right. So you guys all know you have an exam on Thursday, right? So since we're a little bit behind schedule, I think we're just going to skip the unit on biotechnology. So don't worry about that. I'll send out an email announcement, and I'll probably try to update the schedule too. Does anyone have any questions for now? Yes.
Are you gonna put out like one of those study guides that you had last time?
I will definitely try to.
Thank you.
It's a quiz or it's a quiz?
Exam. Yeah, exam 2 is on Thursday. The last day before spring break. I did that on purpose so you guys don't have anything to worry about for this class over spring break.
So it's gonna cover, everything after the life reaction session.
Yes, it will cover everything since the last exam, which begins at the Calvin cycle of photosynthesis and goes up until whatever we finish on Tuesday.
So it's not the whole chapter, it's from Calvin side.
Yeah, because we had the light reactions on the first exam, so I'm not going to test you on that twice. Yes.
You said it's everything from the Calvin cycle until today and Tuesday?
Yes. So whatever we finish at the end of lecture on Tuesday will be the last thing on the